Okay, our next talk is from Drerup. Are you here? Yes, okay. The title is Actr10, a component of the dynactin complex, regulates retrograde mitochondrial transport in axons.
Hi, thanks so much for coming to listen to my talk. I'm going to tell you a little bit about some of the work I've done as a postdoc in Alex Netraporic's lab at OHSU, where we've been really working to identify regulators of unidirectional transport in axons. And the protein I'm specifically going to tell you about today is Actr10. So axonal transport is a critical process for the formation and the maintenance of the nervous system for neural connections. So this process carries proteins and organelles from the neuronal cell body towards axon terminals, or in the opposite direction, from the axon terminals back towards the cell body. So this process is really interesting just from a cell biological perspective because of the way it's actually organized. So axons have microtubules that have a unidirectional polarity. So the microtubules are situated plus end out, and the minus ends are situated towards the cell body. And this is important because the motors that carry cargo throughout the axons, they only walk in one direction. So kinesin motors, which there's an entire superfamily, it's composed of about 45 different genes with multiple isoforms, they're responsible for anterograde transport, which is plus end directed. And so interestingly to me anyway, and I think to the field, is that all retrograde transport, which is transport from the plus ends of microtubules back towards the cell body, is accomplished by a single motor protein complex called cytoplasmic dynein. So I guess what I hope you can gather from that is it's really fascinating to think about the array of cargoes, only some of which are listed here, that rely on dynein to be moved towards the minus ends of microtubules to be properly positioned in axons for their function. I wanted to tell you a little bit about the dynein-dynactin complex. So the core dynein motor is shown here. It's composed of two heavy chain containing domain proteins that have microtubule binding domains. And these can then bind to microtubules and walk along them carrying cargo. In addition to these heavy chain domains, they also have a complement of other proteins on this core complex, including light chains, intermediate chains, and light intermediate chains. So in addition to this already large complex, dynein is oftentimes bound to dynactin, which is shown here in shades of purple. Dynactin binds to the core dynein motor through interaction of P150 with the tails of the intermediate chains on dynein, and that will become important for some of the later experiments I'll tell you about. So again, we're interested in how this motor binds to an array of cargo. And so in order to get at that, we decided to use forward genetics. So we did a forward genetic screen looking for mutants that have axonal phenotypes indicative of impaired retrograde cargo movement. So I wanted to tell you a little bit about the system that we're using. So this is a picture of a 5-day-old zebrafish larvae that's expressing EGFP cytoplasmically under the control of the NeuroD promoter. So for most of my images, when the axons are in green, it's because we use this transgenic for pretty much everything. And so within this larvae, we're actually specifically looking at the axons of the lateral line system for a number of reasons. So the lateral line system at this point needs no introduction. It's a mechanosensory system in the fish. It's composed of these sensory organs that lie along the trunk that sense water movement, and then transduce a signal to the afferent axons that innervate them. So these axons extend from this posterior lateral line ganglion situated right behind the ear, and in the longest instances stretch all the way down the trunk of the fish into the tail. If you look at the innervation of these neuromass sensory organs, you'll see that it's typically a nice basket-like structure. We're using this system for a number of reasons, one of which it develops early. In addition, these axons are extremely long, which makes them susceptible to interruptions in transport. And finally, they're superficially localized, which makes analyzing them in an intact animal very easy. So as I said, we're looking for mutants that have disruptions of retrograde cargo transport. So what do you look for? That's always the question. So we reason that if anterograde transport's intact, cargo should be transported from these cell bodies into axon terminals. But if you lose the ability for retrograde transport to occur, those cargoes should accumulate here, creating axon terminal dysmorphologies or swellings. So that's particularly what I'm screening for. So one of the mutants that we found in this screen is shown here, and it's a loss of function mutation in Actr10, which if you remember from the last slide is a part of the dynactin point-to-dyne complex. So when you look at this mutant generally, it develops rather normally. Axons of the posterior lateral line system specifically extend all the way into the tail. But if you look at the innervation of any of the sensory organs along the trunk, you have these really large axon terminal swellings. So we wanted to know if these swellings were actually resulting from accumulation of a particular cargo. So I did an array of immunolabeling experiments looking to see if any cargoes that we were interested in accumulated there. In addition, I did some mosaic labeling of different cargoes using protein overexpression tagged with fluorescent proteins. And to date, the only cargo we've seen that accumulates in these axon terminals is mitochondria. So what I'm showing you here are wild type on the top and ACTR10 mutant on the bottom, axon terminals. Again, the axons are in green. And on the, left, the mitochondria are labeled with tag RFP, and this is just shown in white on the right. And we did this through a mosaic label using a mitochondrial targeting sequence tagged with tag RFP. So you can see that in the wild type, you have a nice array of mitochondria in these axon terminals. In the ACTR10 mutants, you have this very large focal accumulation of mitochondria in the swollen axon terminals. So this tells us that mitochondria are accumulating in this location, and potentially this is due to interrupted retrograde mitochondrial transport. So why is this exciting? This is exciting for a number of reasons, but to me, the most exciting thing is that we really don't know how mitochondria are unidirectionally moved in axons, so they're positioned at the right place at the right time to perform their functions, which include things like buffering calcium for synaptic activity and producing the ATP necessary to run ATPase-dependent sodium-potassium pumps to repolarize neurons after an action potential. So we wanted to know if indeed loss of function in ACTR10 was resulting in impaired retrograde mitochondrial motility. So to answer this question, we used our in vivo imaging assay where we can look at the motility of cargoes in lateral line axons in intact zebrafish larvae. We can inject DNA, plasmid DNA, that encodes particular things like, for instance, in this case, a mitochondrial targeting sequence tagged with tag RFP. This DNA is mosaically inherited in the developing embryo, and we can titrate this dose such that we can get expression of this mitochondrial targeting sequence in a subset of lateral line axons. So we can tag mitochondria with tag RFP, and then we can watch them move in vivo in an intact animal. So this is a movie showing you mitochondria that should be playing in a minute. You can see anterograde mitochondrial movement as well as retrograde mitochondrial movement. Retrograde is from this direction to here. The scale bar is 10 microns just for your reference. So we can take these movies for wild type and mutant, and then we can analyze them with chymograph analysis to look at transport parameters for mitochondria. So chymographs basically follow the brightest point along a defined line, which is the axon, and then the traces represent the movement of these organelles. So you can see, for instance, in the wild type here, we have anterograde traces going in this direction, and the retrograde mitochondrial traces are in this direction. Now, what I hope you notice when you look at the ACTR10 mutant chymograph is that while we have anterograde traces, we lose retrograde mitochondrial transport. We can quantify this by looking at the number of mitochondria moving in either direction. If you look in the anterograde direction, you see no difference between wild type and mutant in the number of mitochondria moving. And when you look in the retrograde pool specifically, you see this strong reduction in the number of mitochondria that can be moved in the retrograde direction in the absence of ACTR10. Now, another way to look at this is to look at population dynamics, which just takes into account what the pool of mitochondria is doing in the axon. And what I hope you'll notice is that compared to wild type, in ACTR10 mutants, you actually have a slight increase in the proportion of mitochondria moving in the anterograde direction and a strong decrease in the proportion of mitochondria moving in the retrograde direction, arguing that we lose mitochondrial retrograde transport in the absence of ACTR10. So this is extremely, extremely compelling, but we were a bit concerned because there's a little bit of evidence in the literature from one paper that shows that if you lose ACTR10, it indicates that you might actually destabilize the dynactin complex, which would in turn impact all dynactin-dependent cargo motility. Now, if you remember what I said earlier, other cargoes were normally localized, so we had a good feeling that dynactin function was intact, but we wanted to get more at whether ACTR10 mutants were actually going to phenocopy loss of dynein or dynein-dynactin function. So we did a number of experiments I can't tell you about today because of time, but one I'm going to tell you about is the latest, which is using previously existing dynein-dynactin mutants and comparing their phenotypes to ACTR10 loss of function. So we're really lucky that a lot of wonderful mutants exist already for these components in the zebrafish community, and they readily share them with us. So we can look at loss of dynein heavy chain, or we can look at loss of P150 and compare those phenotypes to ACTR10 mutants. And as I told you earlier, dynactin relies on P150 to interact with the dynein core complex. So if you lose P150, it should be reminiscent of loss of dynactin stability. So first I'll show you what dynein heavy chain mutants look like. We cross this mutant into a neuro-DGFP transgenic, and what you'll notice when you compare it to wild type is the axons are extremely truncated and they're actually degenerating. Similarly, if you knock out P150, you get a similar phenotype. So a P150A mutant already existed in the community, but what we found out was in the recent genome build that there's a P150B. So we generated a knockout using CRISPR-Cas technology. So when you look at P150AB double mutants, you have a phenotype. P150A or P150B mutants alone do not. So this shows us that the axonal phenotype in loss of dynein or dynactin function is very different than the gross phenotype in ACTR10 mutants. But we wanted to know if loss of dynactin stability, how that affected mitochondrial motility and mitochondrial localization. So we specifically looked at the localization of mitochondria in P150AB double mutants and compared it to wild type. And what you'll notice is that mitochondria do accumulate in these axon terminal swellings when you lose dynactin completely. But the mitochondria are obviously very clearly unhealthy as they've fragmented here. And we can again use our in vivo imaging assay to look at the motility of mitochondria in this P150AB double mutant. And what you'll notice is if you look at the proportion of mitochondria that are stationary or moving the anterograde or retrograde direction, in the single mutants, you have no effect. But if you lose P150, both isoforms, you have a strong reduction in almost all mitochondrial motility, which is again very different from loss of ACTR10 function, where you actually have an increase in anterograde transport. So this argues to us that ACTR10 depletion does not phenocopy loss of dynein or dynactin. So does ACTR10 actually serve a role in attaching mitochondria to this complex for transport? To get at this, we turn to mitochondrial fractionation. So we can fractionate mitochondria from wild type and mutant larval pools and subject these lysates to roughly increasing speeds of centrifugation to pull down a fairly clean, rough mitochondrial pool. And then we can run those extracts on a Western blot and probe for components of the dynein-dynactin complex. So when we look at the input, you'll notice that there's equal levels of P150 in wild type and ACTR10 mutant pools. This is similar in the heavy fraction. But when you look at the mitochondrial fraction, you'll notice that there's a strong reduction in the amount of P150 in the ACTR10 mutant pool, indicating that when you pull mitochondria down in this fractionation assay, in the absence of ACTR10, you're not able to pull down the dynactin complex as well. Now, importantly, this is not because P150 is being degraded, as it's just shifting over to a lighter fraction. So this tells us that ACTR10 is essential for binding mitochondria to the dynein-dynactin complex. The bottom is a mitochondrial loading control. So in summary, I've told you today that ACTR10 is important for linking mitochondria to the dynein-dynactin complex, and this is in turn necessary for retrograde mitochondrial transport in axons. My continuing work is going to focus on further dissecting the mechanism of this interaction. So we know we've defined the binding domain in ACTR10 necessary for interaction with the dynactin complex. We still have yet to figure out how ACTR10 is actually binding to mitochondria. It doesn't have a membrane domain or membrane-associated domain, so it's likely interacting with partner proteins for this process. We're taking a candidate gene approach as well as doing some mass spec analysis of interactors to identify the proteins working with ACTR10 in this process. In addition, I'm hoping this gives us some insight into how this interaction is regulated. And I'm also really interested in the difference potentially in function for this protein in sensory versus motor neurons. So with that, I want to thank the Netchaporic Lab. It's been a wonderful place to do a postdoc. And as well as the current members, our former members, including Sarah Lesk, who was a technician for us and is now in the greener pastures of Utah. And I'd also like to thank our collaborators, especially Adam Miller, who was instrumental in cloning the mutation in ACTR10. And with that, I'll take any questions. My email address disappeared, but feel free to email me. I'm starting my lab this fall. So if anybody's interested in pursuing these questions, I'm taking motivated volunteers. Thanks.